Hello, dear friends. Welcome again to one more session in molecular biology. I am Dorka Beri, Assistant Professor in Biochemistry, giving you the details about the genetic code. The genetic code is one of the important topic in the protein synthesis, and watch till the end for complete understanding of the topic. Genetic code is the relationship between the nucleotide sequence on mRNA and amino acid sequence in the protein. Or we can also give the definition of genetic code like this: collection of bases that correspond to amino acid, the start codon and stop codon signals for the translation process. Then what is a codon? Let's see. Codon is a hereditary unit of the genetic code. We have seen genetic code definition, which is the relationship between the nucleotide sequence on the mRNA and the amino acid sequence on the protein. Now the codon is the three adjacent nucleotides on the mRNA which specifies a particular amino acid start codon or stop signal is known as a codon. What is a codon? A three nitrogen bases on the mRNA. If this is the mRNA, three nitrogen bases, imagine GGG, are present, which specifies a particular amino acid, or they may also specify a particular signal. For example, if this signal AUG is there, it indicates the starting of the polypeptide chain, or else they may also indicate a signal to stop the translation process. Okay, so the genetic code is nothing but three adjacent bases on the mRNA strand which specifies a particular amino acid start codon or stop signal required for the translation process. Remember, the sequence is three adjacent nitrogen bases side by side. And what is anticodon? Anticodon is also three adjacent nitrogen bases, nucleotides, which are present on the tRNA, which is complementary to the codon sequence, is called anticodon. So where the codon is present on the mRNA strand. Where the anticodon is present on the tRNA. Do you remember in tRNA where it is present? If you remember correctly, the tRNA structure, it is a clover leaf like structure having 5' and 3', and it has the loops like amino acid acceptor arm and T-cycle loop, DHU arm and anticodon arm. This anticodon arm contains anticodon. Three nitrogen bases are present which are called as anticodon, and this anticodon is complementary to the codons which are present on the mRNA strand. Now it is clear definitions about the codon and anticodon. Codon is three adjacent nitrogen bases which are present on the mRNA. Anticodon is three adjacent nucleotides which are present on the tRNA molecule. Okay, this genetic code was first worked out by Nierenberg and Corana in the year 1961 to 66 using cell-free extracts, cell-free protein synthesizing system, and for which they got Nobel Prize. They were awarded with Nobel Prize also. Let's see the characteristics of genetic code one after another. The genetic code is a triplet code. What do you mean by triplet code? It is the three nucleotides which are present on the mRNA. Okay, this is the mRNA strand on which the nitrogen bases are present continuously. For example, for our understanding, it is a triplet code, three nitrogen bases. That is why it is called as triplet code. And this triplet code is present on the mRNA strand from 5' to the 3'. How many codons are present? Total codons in the genetic code are 64 codons are present in total, out of which 61 codons are called sense codons and 3 are called nonsense codons. That means 61 codons, they code for an amino acid, they code for amino acid, they will code for start signal, but 3 codons, they do not have any amino acid. That is why they do not have meaning. So usually they are called as termination codons. These termination codons are UAA, UAG and UGA. These three are called nonsense or termination codons. And as we are discussing with the nonsense termination codons, we will also discuss with the initiation codon. What is the initiation codon? The initiation codon is nothing but AUG. AUG is the initiation codon. This initiation codon AUG, it encodes methionine in case of eukaryotes. In case of eukaryotes, it encodes methionine. In case of prokaryotes, it encodes formyl methionine. That means the formyl methionine is the initiating amino acid which is present in the prokaryotes, and in the eukaryote it is simply methionine which initiates the translation. Total how many codons are present? Total 64 codons are present. So this gives you a clear idea about the genetic code. The genetic code contains a total 64 codons. Four major boxes are there, and here you can write the genetic code. This is the first nucleotide which comes on the left side, and the second nucleotide, this is the second nucleotide which has to be written, and the third nucleotide is this one, UCAG. Likewise, for the second box, you again last third letter is UCAG. The first letter is left side, the second letter is the middle one, third letter is the end one, which is always UCA followed by G. And this genetic code, if we see this genetic code, there are amino acids which contain more than one codon. For example, if we see this phenylalanine, it has two codons, UUU and UUC. For example, leucine, it has UUA, UUG, CCU. This entire box, it is total 6 codons are there for leucine. Likewise, isoleucine has three codons. So here we'll see that all the 20 amino acids are mentioned here. Many amino acids have, almost all the amino acids have more than one codon except methionine and tryptophan. And these are the amino acids which contain two codons each. What are they? Phenylalanine, tyrosine, histidine, glutamine, asparagine, lysine, aspartic acid, glutamic acid, and cysteine have two codons each. And only one amino acid is there which is encoded by three codons, isoleucine. There are four codons for about five amino acids, namely valine, proline, and threonine, alanine, and glycine. Okay, this box entirely proline, threonine, alanine, valine, and glycine. They are encoded by five codons, and there are six codons for three amino acids, namely leucine, serine, and arginine. Leucine we have seen. Here is the serine, two amino acids, and there are again four amino acids are present, serine and arginine. For these three amino acids, there are six codons each. That means the genetic code, it says that it is redundant. That means more than sufficient number of codons are present for each amino acid. I told you before that the initiation codon is AUG. AUG encodes methionine in eukaryotes and formal methionine in prokaryotes, and these three are the nonsense codons. 61 codons are there for 32 tRNAs. In order to carry the amino acids encoded by 61 codons, there are 32 tRNAs which are required. Let's also see some of the other characteristic features of genetic code. The genetic code is universal genetic code. That means in all the organisms, if AUG is there, it encodes only methionine. If CCC is there, it encodes only proline. That means the genetic code is same in all the organisms, starting from bacteria or any organism, it is similar, except the mitochondrial genetic code, which differs slightly from the standard genetic code. And the genetic code is collinear. That means the codons are present linearly on the mRNA strand. And the genetic code is certain. That means it is unambiguous. Unambiguous means without any confusion. If the genetic code is there, for example, CCC is there, it codes for proline amino acid. For example, if GGG is there, it codes for glycine amino acid. Any codon is, it either codes for amino acid or it codes for start or stop signal. And the genetic code is non-overlapping genetic code. Non-overlapping genetic code means the genetic code, this is one codon, the next codon is this. The next codon codes for glycine. Okay, methionine, CCC codes for proline, and GGG codes for glycine. It is understood, that means if it is the overlapping genetic code, it would be like this. AUG is one codon, UGC is another codon, GCC is another codon, CCC is another codon. Likewise, it goes on like this. But the experimental proof due to mutations, mutational experiments were done which says that the genetic code is non-overlapping genetic code. Then the another characteristic feature is the genetic code is redundant or it is degenerate. That means more than sufficient number of codons are present for amino acids. Except for methionine and tryptophan, all the other amino acids contain more than one codon. We have seen that leucine contains 6 codons, arginine contains 6 codons, isoleucine contains 3 codons. That means the genetic code is redundant or it is known as degenerate genetic code. To explain the property of the degeneracy, a hypothesis was proposed known as Wobble Hypothesis, which was proposed by Crick. Francis Crick proposed Wobble Hypothesis to explain the process of redundancy of the genetic code. According to this Wobble Hypothesis, what it says is the 5' anticodon base is unstable and it is called as wobble base. What is 5' anticodon base? Okay, if we see these figures, this is 5', that means this is 5' anticodon. Actually, this is anticodon. This tRNA for this, it is anticodon. Then which is 5' anticodon? This last one is the 5' anticodon. This base is 5' anticodon base. So this 5' anticodon base is unstable on the tRNA and is usually, it recognizes more than one amino acid. And as it is unstable, it is usually replaced by other bases like inosine, which facilitates the binding to more than one codon by the tRNA. The same tRNA would be there, but it can recognize more than one codon. Depending on the wobble base, each tRNA can recognize one codon, two codon, or maximum it can recognize 3 codons also. That means a tRNA can recognize maximum 3 codons, or a tRNA can recognize 2 codons, or a tRNA can recognize only one codon depending on the type of nucleotide occupying the wobble base. This is the example for the wobble base. This is wobble base. G is wobble base occupying, and this is the 5' end of the anticodon. This is the 3' end of the codon. The 5' end is wobble base. It recognizes G, it recognizes C, and also G can also recognize U. Okay, due to this, what happens? The same anticodon, it can recognize codon ending with C or codon ending with U. Did you understand? It can recognize codon ending with C or it can recognize codon ending with U. Okay, depending on the wobble base, the number of codons recognized would be C. Okay, this table gives a clear picture of the Wobble base, first nucleotide of the tRNA, and the last nucleotide of the codon is mentioned here. If the 5' anticodon base, that means if the Wobble base is cytosine, it can recognize only one codon ending with G. For example, if we see this one, UUC, it can recognize only one codon and this ends with G. So to recognize this codon, one tRNA is required. And if the Wobble base is G, it can recognize codons ending with U as well as the codon ending with C. We have seen in our previous case like this. This is the Wobble base. This is the Wobble base which recognizes codon ending with C, or it can also recognize a codon ending with U. That means if G is the Wobble base, it can recognize two codons. If uracil is the Wobble base, it can recognize two codons, one codon ending with adenine, another codon ending with guanine. If inosine is the Wobble base, it can recognize maximum three codons, one ending with uracil, another ending with cytosine, another ending with adenine. For example, if we see these are the codons, imagine GGC, GGA, these are the codons, and if this is the anticodon, this is 5' end and 3' end of the anticodon. If inosine is the 5' anticodon base, that means if it is the Wobble base, it can recognize GGU ending with U. It can recognize codon ending with C. It can also recognize a codon ending with A. That means this tRNA, this particular tRNA containing CCI anticodon can recognize a maximum three codons. That was explained by Crick in the Wobble hypothesis. Hope it is clear to you. Okay, thank you for listening. Hope it is you understood very well, and thank you for watching till the end. And if you like the topic, please like, share, and comment after listening. And don't forget to subscribe. Thank you.